This conference will now be recorded.
Okay. So first presentation by Dr. Jiaoxin Song from Chinese Academy of Agricultural Sciences, China. And the title of presentation is Maltose and Totally Impermeable Film Enhanced Suppression of Anaerobic Soil Disinfection on Soil-Borne Pathogens and Increased Strawberry Yield. More.
Hello, everyone. Can you hear me?
Yes, we can see your slides. You can start.
Okay, thank you. Good morning. Welcome everyone to attend this conference. I'm Zhaoxin Song. I come from the Institute of Plant Protection, Chinese Academy of Agricultural Science. The title of my presentation is Enhance the Effect and Economic Feasibility of Anaerobic Soil Disinfestation combined with carbon sources and cover types in controlling the strawberry soil-borne disease. My presentation includes these 5 parts. First, the background. Strawberry fruit have a bright appearance and delicious flavor and are widely welcomed by the consumers all over the world. With the continuous cultivation of strawberries, there is have a high risk of the soil-borne disease, such as the strawberry root rot. This kind of disease characterized by the drought, the fertility, high temperature, and the reproduction. The soil-borne disease can reduce the strawberry yield from the 30% to 6%, and the overall loss depending on climatic and other conditions. With these problems, we need to find the solution. Soil disinfestation is a good choice. This technology can effectively control the fungi, bacteria, nematodes, weeds, underground pests in the soil, and at the same time can significantly increase the crop yield and quality. We always can choose the chemical disinfestation and the physical technologies. Nevertheless, the main fumigants such as chlorpyrifos and 1,3-D also pose a potential risk to the health of the fumigators and the surrounding environment. Due to increasing regulations and restrictions, we need to find the new technologies as the alternatives for this kind of chemical fumigant. Anaerobic soil disinfestation, ASD, is considered as an environmental friendly and non-chemical technology for pre-plant disinfestation. This technology was reported to be used in Japan at the beginning of the 20th century. And the ASD generally has provided similar levels of broad spectrum control on the soil-borne disease in many kinds of soils. The main steps of ASD technologies, including adding a certain amount of organic carbon source to the soil, irrigating water to soil, and using a conventional agricultural plastic film to keep it airtight for more than 3 weeks. While the mechanisms of ASD are not completely understood, they appear to be a combination of changes in the soil, a microbial community combination, the production of volatile organic compounds, and the generation of critical anaerobic conditions in the soil. The variety of materials and options for ASD application, including the carbon sources, the soil temperature, and the plastic taps, influence the effects of pathogen suppression and disease control. The control effect of ASD on the target is very different. The control effect on the bacteria is between 59 to 94%, the nematodes between 15 to 56%, and the weeds between 32 to 81%. The objectives and the outlines of this research. Based on the scientific problems, a variety of carbon sources and carbon types need to be tested for ASD in efforts to maximize efficacy, reduce material and application cost. Due to potential for past soil cropping system interactions, ASD efficacy and suitability tend to be depend on the specific cropping system and its paths. The objectives include first, determine the optimal experimental conditions with orthogonal design and review the suitability of films for ASD. Second, indicate the mechanisms and ecological benefits of ASD technology by HDS to monitor the shift of microorganisms in the soil. Third, evaluate the application effect of ASD to control soil-borne disease in a strawberry production system. This research contains based on optimizing ASD parameters, including the soil temperature, the soil type, and the carbon sources in vitro to improve ASD technology's stability. 2. Confirming the impact of ASD parameters on the control of plant pathogens and on the soil quality by pot experiment in the greenhouse. 3. Validating the effect of ASD parameters in the field on a strawberry production system to expand its adoption. The anaerobic soil disinfestation, we carried out the optimal design to optimizing ASD parameters. This part, we choose air temperature, the soil moisture, and the carbon sources as the factor. Based on the result, we carried out the current taps experiments to validating the different kind of taps. Here, we choose 3 kinds of film: the virtual impermeable film, the polyethylene film, totally impermeable film, and based on the laboratory result, we carried out the field trials to evaluate ASD as a chemical alternative technology. In this part, we collect the data, include the soil temperature, the fungal pathogens, the soil quality, the microbiota, and the strawberry growth. As end, we give a conclusion as the ASD as an environmental friendly disinfestation technology for the strawberry growers. Part 1. Combined effect of metals and totally impermeable film, TIF, enhanced suppression of anaerobic soil disinfestation on soil-borne pathogens. First, let's see the materials and the methods for the orthogonal experiments. First, the soil prepared. We collect the soil, the fresh soil from the strawberry greenhouse in Beijing, Changping district. Here we give more details about the soil physical and chemical parameters. And we use the orthogonal experiment L27. 15 gram soils were added in a 20 ml head space well and sealed with the cap. The soils were incubated for 21 days. After that, the soil was totally mixed and sampled. We choose 3 factors: air temperature, air moisture, and carbon sources. Each factor descend 3 different levels. At the same time, we used the untreated control based on the different kind of soil moisture. We did the biocide analysis focused on the fusium and the Phytophthora, used the select manner, and the data analyzed based on the one-way ANOVA test. The materials and the methods for the cover types, we choose 3 different films, include the VIF, the PE, and the TIF. We carried out the port experiments, the soil with pathogens from the field outside, and we added 5% maltose as the carbon source for ASD, the PET wave, and also we used the water irrigation and untreated as the control. Each treatment includes 4 replicates and lasting for 21 days. After that, we take the soil samples and did the biocide and data analysis based on one-way ANOVA analysis method. The oxygenal experiments results showed ASD on the fusium and photophosphora showed the different important. For the fusium, the carbon source is important than the soil moisture than the air temperature. But for the Phytophthora, the soil moisture is important than carbon source than air temperature. Also, the optimal combination of ASD was 28 degrees centigrade air temperature, 30% of soil moisture, and used maltose as a carbon source. At the same time, we can see the soil moisture and the carbon source were statistically different significantly according to the ANOVA test for ASD in control, the fusium and the photophosphora, and also the factor A and factor B uncontrolled fusiums have their statistically significantly. The effect of ASD treatments on the numbers of colony of fusium combined under the multiple combination test results showed the group 8, group 5, group 6 have the higher efficacy on the fusiums and the reductions higher than the 90%. So based the result, we choose the 20% of soil moisture and the maltose as factors for the following field trial, different kind of types. The cover types result showed TIF showed a higher effect on fusium and Phytophthora than the PE and VIF based on the reduction rate. And also, we can see there is an interesting result. If we just use the water but no organic amendment in the soil, ASD have no result on the fusium and Phytophthora. However, water and air treatment showed significantly increased the colony of fusium. Also, the rate of suppressions on the Phytophthora was just 51%. This result suggests that ASD using metals as a carbon source requires plastic film cutting measures to ensure the efficacy of soil-borne disease. Okay, the main conclusions for this part. The 3 influence factors showed the carbon source is more important than soil moisture than the air temperature, and the use of maltose as a carbon source is necessary to ensure the effectivity of ASD. ASD using maltose as a carbon source requires the plastic film carbon measures to ensure the efficacy. Based on the laboratory results, we selected ASD, used maltose, and sealed with TIF for the following cover types. The part 2, ASD use maltose and totally impermeable TIF have the potential to replace chlorpyrifos for soil-borne disease in the commercial strawberry production system. For these field trials, we selected 2 locations to carry out in the strawberry greenhouse. This greenhouse is have long history for the strawberry plant history. We carried out different kind of soil treatment and the soil fumigation lasting for 21 days. After removing the taps, and we take the soil samples at 2 times, the 30 days and 120 days after fumigation. We took the samples back to the lab, do the soil quality analysis. This part focused on the soil fungal pathogen, and the samples from the trial 1 and the trial 2. The bacterial fungal community and the soil physical chemical parameters based on the trial 2. We also collect the data for the strawberry response in this based on the trial 2. At the end, we did the data analysis. Here give more details about the trials, include the field trials, physical and chemical parameters in these 2 locations, and the main soil quality parameters also give the data for the field trials. The anaerobic soil disinfestation treatments include 5 different treatments in the 2 trials. MEL6 means consisting of 6 tons per acre of metals applied by drip irrigation using 50 tons per acre and covered by TIF for 3 weeks. MEL9 means consisting of 9 tons per acre of metals applied by drip irrigation with 50 tons per acre water and covered the TIF for 3 weeks. The PIC means correctly injected by the machine as the dosage of 0.375 tons per acre, and then covered with the PE immediately for 2 weeks. The SOL means the solarization treatment. CK means untreated control. All the treatments were replicated 4 times and randomly distributed at each site. The method includes soil samples and the strawberry response. The soil strawberry response will focus on the plant growth, the motility, and the yield. First, let's see the soil temperature and the soil moisture result. The soil temperature is one of the very important factors that influence the efficacy of ASD. The average soil temperature decreased as the soil depth increased. With the lowest temperature value, 23 degrees centigrade appears as the 0 centimeters of the trial 1. In field trials, the average soil moisture during ASD was 36% and 37% at trial 1 and trial 2, respectively. After 30 days of soil disinfestation, at least 89% and 75% reductions in fusium and photophosphor as these 2 trials the soil in our research was sandy loam, and the ASD treatment was for 3 weeks. This combination increased the suppressions of ASD for the pathogens. Naturally, after 120 days of soil fumigation, the colonies of fusium and photophosphor recovered. Based on the HTS analysis result, the alpha diversity of bacteria and fungi were not significantly different between these 5 conditions. Also, the Adonis result showed no significant difference between the ASD, SOL, PIC, and the CK treatments. ASD treatments changed the soil microbial, soil bacteria, and fungal community composition, but the combination was not significantly different as the class and genus level. Without FDR correction, the relative abundance of bacteria class Clostridium and Hyalurobella have the significantly increase in ASD, PIC, and SOL treatments compared with untreated. MAL6, MAL9 and PIC significantly increase the relative abundance of bacteria. General Clostridium, the relative abundance of sequences assigned to Fusium treated with MAL9, PIC, and SOL slightly decreased compared with the CK, but the observed difference was not significant. For the soil quality, MAL6, MAL9, PIC, and SOL significantly increased the ammonium nitrogen and electrical conductivity in the soil, but significantly decreased the nitrogen. The ASD showed significantly increased on the strawberry growth as the trial 2. Also, ASD had significantly increased on the strawberry yield with a lower plant mortalities at trial 2, and there was no significant difference between ASD and the PIC treated. For the summaries of this part, ASD can significantly reduce the colony of this toucan or pathogens, and ASD use maltose as a carbon source and covered with teeth, slightly changed the soil fungi and the bacterial community combination. ASD increased the soil nutrients than the PIC. ASD significantly promoted the strawberry growth, reduced the mortality, and increased the yield than the untreated, and showed no significant difference with the PIC. As a conclusions for this research, first, the carbon source and cover types play an important role in the ASD suppression of soil-borne pathogens. Two, the optimal combination is temperature 28 degrees centigrade, soil moisture 30%. The carbon source used maltose, and the ASD treatments use maltose as a carbon source and sealed with teeth, showed greatly suppressions on the Fusium and the Phytophthora. ASD increased the plant yield, soil quality, and shifted the soil microbial communities. ASD can be used as an alternative strategy to control the strawberry soil-borne disease. The acknowledgements. So thank you very much.
At this corner, at the right side corner.
Thank you very much, Dr. Jackson Song. It was an informative presentation. And any queries?
Sure.
I would like to ask one question. So you've added a maltose into the soil, but is that economically feasible? Because what is the price of maltose?
Okay, your question. Can you hear me?
Yeah.
Okay, thank you very much for your question. So you asked me the price of the maltose. Yeah, exactly.
Yes.
The maltose is not very cheap than the other organic amounts. Normally, the price. Let me calculate. Higher than $400 USD higher than $400 USD per ton of the maltose. This price is a little bit higher for the normally economic crops growers, but you know, for the strawberry is a excellent price because the strawberry's price is really expensive in China, especially during the Spring Festival time. The price for the strawberry is higher than let me calculate, almost 11 USD No. Almost 22 USD dollar per kilogram of the fresh strawberry during the spring festival. So this price is really good for the growers. So even they choose the metals as the carbon source, they can carry out the ASD. Thank you.
Okay, thank you very much.
Okay, thank you very much. So next presentation by Dong Yan from Chinese Academy of Agriculture Sciences, China. And the presentation title is Dimethyl disulfide as an effective soil against nematodes in China.
Hello, everyone.
Yeah, hello.
Hello. Good morning, everyone. And I'm Yan Dongdong from Chinese Academy of Agricultural Science. And I'm very glad to share you something about our research, the results on the dimethyl disulfide as an effective soil fumigant against nematodes. And our research focuses on the bioactivity and the efficacy of DMDS. And my presentation contains the following 3 parts. Soil-borne disease and nematodes cause great damage in greenhouse crops and consequently lead to reduction in crop yield. Soil fumigation is a highly effective technique for control soil-borne pests. Soil-borne pests including soil fungi, bacteria, nematodes, viruses, soil insects, weeds, and so on. Soil fumigation has been widely used on strawberry, tomato, cucumber, ginger, and many high-value crops in China. And these are different soil fumigation technologies used in China, including non-chemical and chemical fumigation technologies. And we can see the crop yield and plant growth was greatly improved after fumigation. And many kinds of nematodes could cause plant soil-borne disease, such as rural nematodes, cyst nematodes, stem bulb nematodes, and so on. And rural nematodes is widely distributed worldwide, and there are 4 most common species of rural nematodes, and approximately 23 species of rural nematodes have been described in China, and 75 non-Chinese species have been reported worldwide, and new ones are being reported every year. It can attack more than 2,000 kinds of higher plant species and cause great damage to agricultural production. And this is the nematode infection on tomato plants. A chemical treatment is an effective way to control nematodes. Generally, there are 2 types of nematicides, fumigant and non-fumigants. And these are common non-fumigant nematicides used worldwide, and also including some biological control agent. And some of the nematicides products were just registered in past 5 years. And these are common fumigants used worldwide. Soil fumigants usually give excellent control of nematodes in soil. All fumigants have low molecular weights and occur as gases or liquid. The fumigant can volatilize and the gas diffused into the space between the soil particles and can kill the nematodes. And DMDS is a sulfide-based volatile compound. It is a natural biopesticide. Alien species are the natural source for DMDS production. DMDS was first registered in 2010 for pre-plant soil fumigation by Acma, a French company, and now it is commercially available for vegetable crop to control nematodes and other soil-borne pathogens in many countries such as USA, Korea, Mexico. And DMDS is now under registration in China. And this is the small injection machine for DMDS application in China. This small injection machine can work both in the open field and the small greenhouse in China. And our research results contain the following two parts: the bioactivity and the feed efficacy of DMDS. And in our laboratory experiment, we use 3 methods to identify the direct contact activity and fumigation activity and the soil fumigation activity of DMDS. The bioactivity of DMDS was compared with 1,3-D, a traditional fumigant for nematode control. And from the direct contact activity result, we can see the LC50 of DMDS is 29 milligram per liter, and which LC50 of 1,3-D is 18 milligram per liter, which means 1,3-D has a higher direct contact activity than DMDS. The direct contact activity was determined in the water solution, so the activity is seriously affected by the emulsification efficiency of the pesticide. And both DMDS and 1,3-D has excellent fumigation activity. The fumigation activity of DMDS is quite similar to 1,3-D. And for the soil fumigation activity, 1,3-D seems to has a higher activity than DMDS. And in our field study, 4 dosage of DMDS was set in our experiment, range from 10 to 80 gram per square meter. And the experiment was conducted in greenhouse cucumber and tomato. And the rural index and the crop yield were determined in each treatment. And from the results, we can see the highest rural index were both observed in untreated control treatment. And what DMDS treatment reduced the rural index. And this is the tomato and cucumber root in fumigation and the long in untreated treatment. It is quite obvious that the plant root in fumigation treatment has less roulots. And all DMDS fumigation treatment significantly increased the crop yield compared to the untreated control in both cucumber and the tomato fields. And in the conclusion, in our laboratory study, DMDS shows good bioactivity on nematodes, and the recommended dosage for DMDS for feed fumigation is 10 to 20 grams per square meter for controlling rural nematodes for short-term crops such as cucumber and tomato. And the MDS is technically feasible soil fumigant for controlling nematodes in China. And besides the research on the activity and the efficacy of the MDS, we also carry out study on the compilation use of the MDS. And with the bad smell of the MDS, we also developed new formulations of the MDS to minimize the bad smell. And okay, let's work on my presentation. And thank you for your listening.
Thank you very much, Dong Yan. And any queries? Next presentation by Dr. Ajay Singh from Institute of Technology, Kharagpur, India. And the presentation title is Effect of Treated Soil and Environment.
Are you able to see my screen? Can you see my
Yes,
screen?
we can see your screen.
And can you also
Yes,
hear
we can
me?
see.
Okay, thank you.
Yes, we can hear you.
Okay, so yeah, hello everyone. Myself Ajay Singh from IIT Kharagpur, India. And the title of my presentation today is Effect of Treated Wastewater on Soil and Environment. So, and this presentation includes background of wastewater irrigation, why wastewater irrigation is needed for achieving food security and increasing farm income, and what is its historical aspect in the background of declining water resource base, and which fertilizer value it can add to the soil if irrigated with irrigation, that wastewater irrigation. But the main objective of this presentation is to show the environmental impact, that is negative environmental impact of wastewater irrigation, such as soil and plant contamination, human and animal health, and climate and greenhouse gases emissions. And at the end, I will give some recommendations for future research regarding sustainable wastewater irrigation and development. So these are the well-known global issues. This is not new. The world population is increasing. So world population is currently at 7.6 billion, which is expected to increase to 9.8 billion by 2050. So during the next 30 years, 2.2 billion additional people will require 60% more food. And that 60% more food we have to produce with declining water resources. That is the result of urbanization, climate change, and contamination. And that additional food, we need more than 65% additional water to produce that food. So this is the problem, and that's why we are looking for alternative irrigation sources, alternative water sources. But why we are talking about irrigation? Why irrigation is important to achieve food security? That the additional food which we require by the next 10 years, 20 or 30 years, that is expected to come from irrigated area and not from non-irrigated or rain-fed area. And why I'm telling this? Because see today's figure, see it is clearly shown here that 23% of total crop area is irrigated as of now, but it provides more than 42% food. That is on global scale. And that 42% uh, food which comes from irrigated area, that is expected to grow to 45% by 2030 and more and over 50% by 2050. So we focus on expanding irrigation in additional areas, bringing more rain-fed area under irrigation, and strengthening the current agricultural that irrigation practices. So here you can see that 2/3 of world population currently live in areas that experience water scarcity for at least 1 month in a year. And this is very interesting to see that almost all the world populous area, say for example South Asia, one example, China could be another example, they are facing some sort of water scarcity. So say for example South Asia here, uh, which is a home of around 25% of total world population, and this figure is include that China population is included together with this South Asia population, that will come around 40% of total world population. And we can see in this range, this area experience water scarcity problem ranging between 6 months a year to almost entire year. Here you can see. So wherever population density is higher throughout the world, you can see not only South Asia or China, wherever population density is higher, of course barring a few exceptions, that those areas facing water scarcity. So we need to think about alternative water sources. And 50% of people facing this level of water scarcity. So climate change will further aggravate the situation by increasing temperature and by increasing the frequency of severity of floods and droughts. That in turn will reduce the available water resources further. So here are the key drivers: increasing world population, changing dietary pattern. Changing dietary pattern, what I mean here is that changing pattern with income increasing, people will become more aware and more educated. They will need additional water for their personal, professional, and even crop irrigation. So that water requirement will be higher in that case. So fresh water scarcity already happening. So what is wastewater? Anything which say for example wastewater comes from various sources. So municipal wastewater, which contains 99% liquid plus 1% solid, and domestic effluent, which consists black water and grey water. Grey water includes kitchen and bathroom wastewater, while black water can include irradiated associated sludge. So water which comes from a commercial establishment and institutions, including hospitals, are also considered wastewater, which can, after uh, suitable treatment, can be used for irrigation purposes. So industrial effluent could be another source of wastewater. Storm water and other urban runoff. I'm not today presenting that marginal poor quality water, but that could be also one of the additional water sources if we look for that. So why wastewater? Why we are talking about wastewater? We of course need additional water. We are looking for alternative water sources, but why wastewater? Because wastewater is uh, uh, rich in nutrients. It's a source rich in nutrients, and it's highly productive resource. and more importantly, it is available throughout the world 24/7. It is produced everywhere. If after proper treatment, it can be used suitably. It could be a good alternative for that additional water sources, which we require to fulfill our rising demand in the coming future. And all the essential nutrients required for crop production is available in wastewater. I will show in the coming slides that how much nutrient we can get by irrigating that crops by that wastewater. So with proper management, wastewater use can contribute significantly to sustaining livelihood, food security, and the quality of the environment. And wastewater irrigation is not new. It has already been practiced for long. Say, for example, more than 20 million hectare area of cropland is already irrigated in that already under wastewater irrigation, and that area is continuously increasing due to climate change and the rising water demand. So why wastewater? Also here you can see that water demand from other sectors. See, agriculture is the largest water user and it consumes over 70% of total water sources, developed water sources worldwide. But that figure is going to decrease because of fierce competition from other sectors. So for example, domestic and municipal that sectors that need additional water in the future. So that water availability will be reduced in future, and but at the same time we need additional water to produce more crop. So just for example, if I could give here that in India 83%, more than 83% of developed water resources annually is used for crop irrigation, but that figure is projected to decrease by 69% in the coming 3 decades, that is by 2050. So because other sectors are also demanding more water, so it is not fully industrialized country. Industries are also growing. Urbanization is fast rising there as compared to developed countries. So we should allocate more water to those areas also, leaving very less quantity of water as compared to today's current level. So we need additional source of water. And why wastewater? Because it's a rich source of fertilizer and nutrient which are required for crop production. So for example, if you irrigate, if you allow, if you put 4,000 meter cube per hectare irrigation with wastewater, that could add up to 248 kg nitrogen in soil, and similarly it could also add 276 kg potash and 96 kg phosphorus, and similarly other also micro major nutrients are available in wastewater. But of course, this amount of fertilizer or nutrient will depend on the concentration of wastewater. So more the higher the concentration, higher the value will be added in the soil profile. So for example, if concentration of wastewater is only 18 milligram per liter, it will add only 64 kg of nitrogen. When it increased to 60 kg, 60 milligram per liter, that concentration, it will add 248 kg of nitrogen with the same amount of that wastewater application. So wastewater is good for everything. It is increasing water base. It is increasing water base could be one of the best alternatives because it is available everywhere and all the time, available 24/7. So what is the problem in wastewater? This is the problem. Because 80% of wastewater globally is released to the environment without adequate or in many cases with any without no without any treatment. That is the problem. Even high income countries, you can see only 70% of total waste is treated before being released to the environment, and condition is very worse in lower income and even middle income country, lower middle income countries, where it is treated by 8% or 28% only. So the lower income countries, 92% of wastewater is released into the environment without any treatment. So without proper management, wastewater use poses serious risks of human health and the environment, which will be shown in the next slides. So what is the problem in using wastewater? Improper, inadequate, or no treatment at all. So whatever water, wastewater we are using, say there is a huge, I mean, big misconception that we should treat wastewater only when we want to reuse it for agricultural or for industrial or for other purposes. But this is completely misconception. Even we are not going to reuse that wastewater, we should release it only after proper treatment. Because if, say for example, if we are going to if we are not going to use that wastewater for any purpose, and we release it to the environment, to river or to water body or to anywhere without proper treatment, that will add a lot of pressure, and that will lead a lot of population, that pollution, and may cause various diseases. So basically, poorly treated or no treated, untreated wastewater can cause three types of major problems. That is, one, soil and plant contamination because of accumulation of heavy metals, salts, and toxic elements; altered physical conditions, that is, changing hydraulic conductivity and leaching efficiency of soil, that will further decrease the leaching efficiency of that toxic salts through the soil layer, and that will after accumulation, after some time, it will translocated to plant, and once we use those plants product, that will go to our body, and it will start that harming our system. So, and human and animal well-being, so many diseases caused by pathogens, bacteria, helminths, and viruses, etc., which I will show in the next slide. So contaminants of emerging concern could also one of the very, global concern. Recently, say for example, extensive use of pharmaceutical, pesticides, personal care products, and other toxic organic compounds are the major that threat of that using untreated or inadequately treated waste for irrigation or for other purpose. And third one is the greenhouse gas emissions, that methane, carbon dioxide, nitrous oxide emissions from wastewater. Various studies confirm that that storage and use of that inadequately treated wastewater could lead to greenhouse gases emission, which in turn lead to climate change. So these are the pathogens present in wastewater and their survival time and their related diseases. So pathogens could be bacteria, helminths, protozoa, viruses, and vector-borne diseases if wastewater is not properly treated. So bacteria can present anywhere between 1 to 10 power 10 number in 1 liter of that wastewater, and it could cause, if not properly treated, to diarrhea, cholera, gastroenteritis, arthritis, etc. Similarly, helminths, protozoa, viruses, vector-borne pathogens are also responsible for causing various diseases. So if water is not properly treated. So one thing we can see here, that proper treatment, why proper treatment is required before wastewater is being used for agricultural or for any other purposes. See here, so for example, helminth eggs can survive many years in water and sewage water environment. Here you can see, and even in soil, they are not going to die any further if we are not treating, if we are not doing anything. Once you use that wastewater, not necessary that the helminth eggs are present are going to harm you immediately. They can harm even after years, because their survival time is many years. In that come helminth and this one, and even protozoa can survive for up to 15 to 180 days. So that proper and adequate treatment is very much necessary. So considering these restrictions and environmental impacts of wastewater irrigation, here are some qualitative criteria for wastewater irrigation. So electrical conductivity of wastewater, which is once it is less than 0.7, there is no restriction. So that quality of wastewater can be used for any crop or any soil condition. Once EC value that rises above 0.7 but is still below 0.7, in that case wastewater has some low to moderate restrictions of its use, and once it crosses 3.0, that is high restriction. So that is very selective. Only salt tolerant crops or some light soil in condition can be used. Similarly, other parameters are also there, so TDS, sodium, nitrogen chloride, all those things. So, and the details is given in current by recent publication that is published in Research Conservation Recycling. So viewers are, if interested, you can go through that paper. And if you are not able to get access of that paper, I can send you. So wastewater irrigation, which are the groups which are at maximum risk? Close communities where wastewater is stored, where it is being used, and farmers and their families, of course, they are that major risk groups, and that because they are they who face most that wastewater use, and that from farming to from cultivation that growing to harvesting a crop, and finally consumers. So consumers, why consumers? Because see, that depends. If and most consumers are at at risk mainly because if they eat raw vegetables or raw products which are produced by the use of wastewater irrigation. Say for example, radish and all the that carrot, they are eat they are eaten that raw. So if they are produced with wastewater irrigation that is not properly and adequately treated, that could be a threat to their health and overall being. So how can we minimize the risk to people and the environment? Actually, women and children are the high risk groups. So exposure to vulnerable groups, especially women and children, to partially treated or untreated wastewater requires specific attention. And actually, raising public acceptance and social awareness could also be helpful in avoiding that related negative impacts of wastewater irrigation. So water reuse schemes can fail if planners do not account for the dynamic of social acceptance. Extensive information campaigns and participation by the public are required to build trust and overcome the so-called youth factor. Why this youth factor? Because many people, even if first of all, they don't know whether that produced by using wastewater. So that wastewater is that properly treated or partially treated or no treated at all. Once they hear the word wastewater, they say no, no, no, I am not going to use this product because wastewater is used for that irrigating that crop. So we can see people here and there. If we don't know that where that produce is that grown or where it comes from, then it's fine. So we need that extensive awareness and that some that awareness and some information campaigns for overcoming to this type of misconception. And building capacity and knowledge could also be one of the helpful factor to reduce this negative impact of wastewater irrigation. So campaign building, research and development aimed at improving wastewater management, generate employment opportunities and promote green growth. And most important thing that while we are talking about building capacity and knowledge, so transfer of technologies from developed to developing countries, because developed countries are better equipped to formulate and implement better and advanced technologies. So they are suggested, they are expected to share, they are expected to transfer those technologies if they have any to needy person to developing countries, because technology developing, formulation and implementation are very costly. Maybe that low income countries may not be able to that bear that type of expenses. And the last conclusion and take home message that wastewater increasing worldwide, but the problem is that it huge vast majority release without treatment. That is the problem. So we need an affordable low cost treatment option should be available to treat to adopt that better treatment, so that minimum water or no water, no wastewater could be released to the environment without treatment. and reliable sustainable source of water. It supports a reliable sustainable source of water if judiciously used after proper treatment. So an appropriate wastewater management is essential for achieving the 2030 agenda of our sustainable development goals. That's all about my presentation in a nutshell. That presentation was this is part of my paper which is recently published in Resources Conservation and Recycling. So if you are interested in details and other aspects of the paper which has not been covered here, it is not possible to cover everything of a 17, 18, 20 paper, 20 pages paper. So if you are interested in that paper, I can help you if you are not able to access that paper. So that's all. Thank you very much for your presence. If you have any specific questions regarding this presentation, you are most welcome.
Thank you very much, Dr. Ajay Singh. You can mention the article details in chat so that everyone can see that, can view that URL.
Okay, just 1 minute, just 1 second. Okay, I've given the reference. You can check.
Yes, okay. We have received your link. So interested ones can go through that link and view that article for more information.
Okay, if you are interested, of course, yes.
Okay, so any queries? So next presentation by Dr. Dilek Tekdal from Mersin University, Turkey. And the title of presentation is Bacterial Identification in the Root of Candidate Intergeneric Hybrid of Common Bean.
Yes. I share my screen, so can you see it?
Yes, we can see that.
Okay, now I will start. Hello, everybody. My name is Dilek Tekdal. I am a scientist at Mersin University in Mersin, Turkey. Today I will mention about my a part of our projects, actually a part of our projects. You can see that the name of that parts of that projects. I will mention about the bacterial identification in the root of the candidate intergeneric hybrid of the common bean. So to write my presentation, I followed this section. So first, I will mention about the general characteristics of Borrelia turcica and the Phaseolus vulgaris. Then I will follow the literature review, but not the more details I will give you. I will just for the little details about the literature review and motivation. Then I follow the section 2, it's including the intergeneric hybridization. Then I follow the isolation and the characterization of bacteria. Then I will finish my talk mention about the conclusion. So let me start with give some details about Borrelia turcica. Why we selected this plant species? Because this plant species is the endemic Turkish species. And this unique plant species has a long rhizome, is a hermaphrodite with golden, you can see here the golden yellow flowers and 2 three-seeded legume fruit. But the most important characteristic is that plant species is to have the at least the 2, but mostly 3 and 4 free carpellary ovary. So you can take the much more yield from that plant species. This plant species is located between the southwestern part of Akşehir Lake and the southern part of the Eber Lake in Turkey. But currently this plant species is under the heavy constructions, since the unidentified predators utilize this plant species seeds for its larval development, and excessive agricultural practices also. And due to all of these reasons, this plant species has been cited in the Red Data Book of Turkish Plant Species as a critical endangered plant species, unfortunately. This plant species is self-pollinated and also the perennial herbaceous plant species. So can you see here? So you can see that there are many studies conducted on that plant species, but mostly micropropagation at the mineral matter and alkaloid content of that plant species is mostly conducted. So in our project, we aim to also contribute to hybridization possibilities of that plant species as well. So you can see here the research organization. So firstly, we conducted the classical hybridization techniques, but to analyze the results, so we collect samples the first day of until the 5th day of the pollination for the histological analysis. Actually, we did not aim to analyze the bacterial species. But during our research, we also observed the abnormal root development in the hybrid candidate plant species. So then we aimed to also analyze which bacterial species affected this abnormal root situation in these hybrid candidates. Actually, all these plant species has a very unique characteristics and distinguished from the other members of the belongs to the family of Fabaceae, but there is no any hybridization studies. Actually, during my PhD thesis, I also conducted hybridization studies, not the Phacelis vulgaris, but we used also the Vicia faba. Also in that hybridization studies, what we recognize that also the embryo rescue studies necessary during these hybridization studies. Then in these studies, we aim to use the Phacelis vulgaris now. Also, the trait that provides the multicarpellary gymnasium in Veralia turcica can be transferred to other species in Fabaceae by intergeneric hybridization. In the future, also, we aim to use another members of the Fabaceae family for the hybridization possibilities to analyze these plant species as well. So this study was to search whether gene transfer from Veralia turcica to another species within the same family Fabaceae by classical hybridization. During our studies, also we recognize abnormal root development in the hybrid candidates. So also we secondly aim to analyze the bacterial community in these abnormal roots as well. Phacelis vulgaris common bean and is the one of the legume crops with the word, and also Veralia turcica is Turkish endemic plant species. They were selected for these studies to analyze the possibility of gathering hybrid plants by intergeneric crosses. Actually, we tried to analyze the intergeneric hybridization because this plant species Veralia turcica is only one representative of the genus Veralia. So there is no any species belongs to this genus. So only one genus we can select, so Veralia turcica, and there is no another species in the world as well belongs to the Veralia genus. But we also selected Phacelis vulgaris. Actually, in our studies, we conducted not only the one genotype of the Phacelis vulgaris, we selected nearly the 12 genotypes of the Phacelis vulgaris, and we obtained the seeds from the local breeders, and non-hybrid seeds we also obtained. Phacelis vulgaris seeds we obtained from the local breeders in Turkey for this study. So first we carried out the hybridization. Then we collect the samples and also we analyze this, especially first the histology. We conducted the histological analysis because first we attempted this study and we don't know if there is any incompatibility events between these species, Borrelia turcica and Phaseolus vulgaris. So we need to analyze those histological analysis to confirm that pollen reached the ovary and also the pollination was successful as well. So you can see that the ovary samples, this image belongs to Borrelia turcica, and the other one belongs to the Phaseolus vulgaris. So you can see that, and you can understand from this figure, it's very hard to study with the Phaseolus vulgaris because the pistil sample is very thin, and during this study, so it can be damaged easily, unfortunately. In this study, we selected the Phaseolus vulgaris as a female donor and female parents, and we also used the Borrelia turcica as a male parent donor plants. During the study, we sown the seeds in a vial, including the peat and perlite in equal volumes. You can see the vials, and in the greenhouse, we used the greenhouse in Mersin University until the plant is obtaining. Then we use this. We use the vials because germination can be easily. And also due to avoid any weeds possibilities in these plant species as well. Then when we obtain the plantlets, we transfer these plantlets into the pots, including the red soil, peat and perlite in the equal volumes as well. Actually, this plant species is cited as an endangered plant species. Also, the Turkish Agriculture Ministry is banned to collect this plant species from the natural habitats. So we studied with the botanical garden, Nezat Gökçe Botanical Garden in Istanbul, together in that research. They have permission to collect rhizomes of these plant species from the natural habitat and to sow these rhizomes in the botanical garden. So we studied with these groups, and they sent you can see the flowers of the Borrelia turcica. They sent our these flowers in the sufficient growth stage. So we removed the anthers and we obtained the pollens for the hybridization. But before the hybridization studies, first we analyzed the pollens viability. For these analyses, we conducted the acetocarmine staining. So you can see also we observed the pollens under a fluorescent microscope. So you can see that these pollens, the red pollens, are viable. For histological analysis, we collected the samples from 1st to 5th day of the pollination. For histological analysis, we collected samples into the FPA70 solution. And after that, we conducted the fluorescence microscope analysis. We prepared the samples for this analysis. You can see for the methodology, for the preparation methodology. So when we look at morphological observation, so unfortunately we conducted these studies last year, and unfortunately the African hot weather conditions also occurred in our cities. Mersin is one of the hottest city in Turkey, and many samples affected these hot weather conditions. So we obtained the pods. You can see the draining pods and the yellowing pods, unfortunately. Also, in some samples, also there is no good development of the ovaries inside in that pods. But we obtained also the samples and we analyzed the histological analysis. So arrows indicates that the sample in these samples, the hybrid samples, hybrid candidate samples, pollen tubes growth until the ovary. Here is the ovaries and here is the stylus. We can say that. So we obtained the hybrid candidates, not much more the samples we actually obtained in that from these studies before of the climatic conditions, unfortunately. But we obtained the little hybrid candidate samples. You can see the samples morphology and also the seeds. Then we sounded these seeds into vials, including the botanic garden, but also the climatic conditions occurred, actually they affected negatively in that plant species growing. So we obtained this means that the number means that the genotypes number actually, because we use the 12 genotypes of Phacelis vulgaris. So we only just obtained the hybrid candidates from these genotypes. So you can see here that the abnormal roots developments, and also there is another, the abnormal development in that plant species. Then we also isolate the DNAs for the molecular analysis. So because we don't know exactly these are the hybrids or maybe the pure lines, because we know that from the literature reviews, when we conducted the intergeneric hybridization, so we can also obtain the pure lines. So we also analyze for these plant species hybrid or the pure lines. Actually, our previous projects results, we also designed the SSR primers of the Viralia turcica. So we select the good SSR primers for these analyses, and for the bacterial identification, we use these primers as well. So you can see the isolated DNAs, and also as a result of the SSR analysis. So you can see that this means that the first means hybrids, the other ones is the controls, and you can see the following the numbers, and the last one is the donor plants DNAs. So you can see that these hybrids, also including the controls, female controls, and the male controls, you can see the same bands is similar with female and the male parents. So we think that these plant species could be hybrids, but now currently actually we also studying to design the newly SSR primers to confirm that these samples is really hybrid plants. So we also analyze that which bacterial species can be affected on the roots. So because previous to slides, you show that abnormal roots developments occurred in these hybrid candidates. So we also analyze for which bacterial species could be affected in this abnormal situation of the roots. We obtained the single colonies, then we also isolate the DNAs, bacterial DNAs, genomic DNAs, and also use the actually conducted the PCR analysis and for the sequencing. So according to balance sorting, so these plants bacterial species belongs to the Bacillus and the Microbacterium genus. So we also analyze the phylogenetic analysis using the MEGA program. So you can see that these slides also there is a 3 clades. We also add the Agrobacterium thymophasians sequences in this analysis as an outgroup. So you can see the 3 clades. One is Agrobacterium, the other one is the Microbacterium genus, and the other one is the Bacillus genus. So this phylogenetic tree also confirmed our results as well. So we can also, as a conclusion, we can say that actually these findings of this study will be a source of the breeding purpose of Viralia turcica for future experiments. Also, it is possible to obtain the embryo from cross-pollination between the Viralia turcica and the Phacelis vulgaris, but we are also currently studying about this in deeply. Also, future steps for the understanding the relation between the isolates and the intergenetic hybrids Phacelis vulgaris include analysis of sterophores and morphophysiological characters of the isolates as well. I would like to thank to National Science of Turkey and TÜBİTAK for financial support to this research. And also I would like to thank the Nezat Gökil Botanical Garden for sending the research samples as well. Gralia tursicans flowers they sent us, flower buds especially they sent us. And also I would like to thank my research group, İknur Akça, Aslı Küçük Recep, Mehmet Selim Çetinler ve Rüşt Hatipoğlu. Also, I would like to thank the organization team to give me an opportunity to present my research as well. And I will also would like to thank you all for your attention. Thank you.
Thank you very much, Dr. Dilek Tekdal. And any queries? Okay, so next presentation by Dr. Rahma Jardak from Center of Biotechnology of Borj, Tunisia. And the presentation title is Proteomic Responses of Barley Landraces to Salt Stress at the Tilting Stage.
Yeah, it is visible. Can you please make it in slideshow mode so that the entire screen will be visible? Like, could
presentation?
So we can see the slides.
Do you see my presentation?
Yes,
Hello?
yes, we see the presentation.
Yes. Hello, everybody. So,
Hello, can you hear me? We can.
yes, I hear you. Do you see my presentation?
Yes, you can start. Yes, we can see your presentation. You can start now.
Okay. My name is Dr. Rahma Jardak from Tunisia. I'm assistant professor at the Biotechnology Center of Borj. I will present you today our research on the proteomic response of barley landraces to salt stress at the tillering stage. And this work was done in collaboration with Leibniz Institute of Plant Genetics and Cropland Research in Germany and Luxembourg Institute of Science and Technology. Just I want to make bigger the. Our work concerns cereal, which provide the most important basis for human nutrition and worldwide. But in view of a growing human population and in view of climate change, tremendous efforts are necessary to ensure cereal crop productivity, which was strongly affected by abiotic constraint, mainly drought and salinity, causing decrease in crop yield of up to 60%. Among cereal, barley is ranking fourth in terms of production all over the world, mostly in Europe, with up to 35% of export. And among cereal, barley is usually used for human consumption and feed for animals. Barley is also a model plant for cereal with a low chromosome number, a relatively short life cycle, and a complete sequenced genome. So in Tunisia, barley is ranking second in terms of production, occupies between 34 to 38% of the cereal cultivated area. As for production, 10 to 15% for human consumption and 85% for feed animals. Unfortunately, climatic fluctuation have caused deterioration on barley adaptation capacity. This was due mainly to salinity, which affected 25% of the arable lands and contaminated 30% of the water resources used for irrigation, leading therefore to a degradation process that induces plant growth inhibition. So during the last decade, yield fluctuated from 4 to 13 quintal by hectares, requiring still the import to fulfill the needs. Nevertheless, barley is the most salinity tolerant cereal crop with varying level. It's therefore a pool of key genes useful for determination of effective barley breeding strategy. So in this context, plants have developed in response to salinity, metabolic process that induces adaptation by altering genes or protein expression. During the last decades, genomic and transcriptomics have made a large progress, but correlation between mRNA and protein is not always observed since post-transcriptional modification is of fundamental significance. So we opted for proteomics to investigate on specific salinity tolerance biochemical traits in Tunisian barley accessions. So our objective consists on the use of selected genotypes with contrasted salinity tolerance to carry out physiological and biochemical assessments under different stress conditions in order to perform proteome analyses based on p-dimensional electrophoresis coupled to mass spectrometry, and in order also to perform crossing and generation of double haploid barleys. Altogether will allow the annotation of key biochemical pathways for salt stress tolerance and identification of traits. These traits will be useful for selection of genotypes with enhanced salinity tolerance for direct exploitation of saline soils. So we conducted in our laboratory of plant molecular physiology, researches which consist on genetic diversity and physiological salt assays. And these researchers allowed the identification of 3 barley pairs with contrasted salinity tolerance at different developmental stages. These genotypes are now exploited for barley proteome response analysis at seedling stage, chilling, and maturation to better understand the tolerance mechanisms, to annotate the biochemical correlations, and to identify the key candidates. So we start with the analysis of the mature grain using genotypes with contrasted salinity tolerance at the germination stage. And this analysis concerned the albumin and the globulin fraction involved in cellular processes, germination, maturation, and defense. So the results of this comparative analysis of this fraction highlighted the contribution of genotype-specific protein, such as small heat shock protein, implicated in the protection and the regulation of protein function, as well as abundant protein, including late homogenesis abundant protein involved in the osmoprotection during desiccation. We continued with the analysis of the proteome response to salt stress at the seedling stage. These analyses were carried using hydroponic salt assay and to analyze the root and the leaves. So the results of the root proteome analysis highlighted the contribution of the amino acid metabolism. And for the leaf proteome response, we noted the contribution of the defense protein. Now, regarding the barley proteome analysis at the stilling stage, we used a genotype with contrast salinity tolerance, and we apply salt assay using sodium chloride concentration up to 200 millimolar during 14 days, and the harvest was done after 14 days for the fourth leaves. So the results of this analysis of this assay showed that in the tolerant genotype, we have less reduction of fresh and dry material, as well as tiller number compared to the sensitive genotype. We noted also distinction for the photosynthetic activity in response to salt stress, where for the stomatal conductance we have more reduction in the tolerant genotype compared to the sensitive, while for transpiration and assimilation we have more reduction in the sensitive genotype compared to the tolerant. So this distinction at the physiological level, we continued with the proteome analysis, and so we perform total leaf protein extraction. So after extraction and solubilization, the quantification showed protein concentration ranging from 7 to 10 microgram per microliter. These extracts were then checked by SDS page and showed similarity in profile. The extracts were then loaded for bidimensional electrophoresis using immobilin dry strip gels of 17 centimeter with pH ranging from 4 to 7. So after strip equilibration, SDS page staining and image acquisition, we successfully performed 24 2D gels, and we matched 311 spots between group of all technical replicates. So the comparison between control and stressed proteome profile showed within the sensitive genotype higher number of differentially abundant spots compared to the tolerant, while in the tolerant we have a higher number of unchanged spots compared to the sensitive. This result noted the distinction between the tolerant and the sensitive proteome. Moreover, the combined comparison between proteome allowed the selection of 74 differentially abundant protein, and these differentially abundant protein were classed in cis groups, where the most important classes was represented by unchanged spot in the tolerant and reduced spot in the sensitive. These differentially abundant spots were then cutted and treated and subjected to mass spectrometry. So after digestion with trypsin and mass acquisition, the identification was performed by MALDI-TOF and by Mascot software by searching in different databases. So we could identify 38 salt-responsive protein in the 4 leaves. Moreover, these proteins were subjected to annotation, and we could found that these proteins are involved in 10 biochemical pathways, where photosynthesis and redox regulation were the most important biological function. On the other hand, these proteins were subjected to hierarchical clustering analysis to select K candidates with the aim to group a protein with similar profile, either under control and or salt stress conditions. So we could note 5 classes, and among the class represented by the abundant spot, abundant protein in the stressed plants of the tolerant genotype, we could select 5 proteins, selected 5 proteins candidates, which were involved in signaling, nucleic acid binding protein, protein synthesis, redox regulation, and photosynthesis. So among these biochemical pathways involved in barley response to salt stress at the tillering stage, we suggest that signaling, nucleic acid binding, protein synthesis, carbohydrate metabolism, amino acid metabolism, energetic metabolism, as well as redox regulation and defense would contribute to salinity tolerance. So in conclusion, the comparative analysis of the 4th leaf proteome of the selected contrasting landraces in response to salinity at the tillering stage revealed significant differences between control and salt-treated proteome, as well as between stressed proteome of both genotypes. We also revealed that salt tolerance may be attributed to enhanced signaling, nucleic acid binding, protein synthesis, redox regulation, and photosynthesis. So our research will be also continued by characterizing the K candidate in order to explore their accurate function in salt tolerance of the Tunisian barley land races. We will investigate also proteomic responses of our barley land races to salt stress at the maturation stage in order to select the biochemical marker for salinity tolerance. And thank you for your attention.
Thank you very much, Dr. Rahma. Any queries?
Thank you.
A next presentation by Anahit Alexandrian from Ministry of Environment of the Republic of Armenia. And the title of presentation is Main Sources of Dioxins Generation at Metals Thermal Treatment. Dr. Anahit, are you ready with your presentation? Dr. Anahit, can you hear me? Dr. Anahit, are you available now? Can you hear me? Hello, so Dr. Anahit is unavailable now. We'll move on to the next presentation by Dr. Wafa Muhammad Abdullah Hikil from National Research Center, Egypt and University of Tabuk, Saudi Arabia. Dr. Wafa, are you available and ready to present your talk?
Yes, I am here.
Okay, the title of presentation is Egyptian Standard Specifications for Drinking Water, Parasitic Point of View.
So please, how can I share my screen? Because it is not available
Okay.
to me. Okay.
Can you try now?
Okay.
I have made you the presenter.
Yes. Are you see my my presentation now?
Yes, we can see that.
Okay. Hello, everybody. It's a pleasure to me to join for second international conference on environmental sustainability and climate change. My name is Wafa Hikal. I'm associate professor for parasitology lab, water pollution research department, National Research Center, Egypt. Today I'll talk about Egyptian standard specifications for drinking water parasitic point of view. Water is life, and access to clean drinking water is a fundamental human right. For human health, drinking water standards and guidelines have been developed to ensure their quality. The World Health Organization has considered access to safe drinking water a basic human right to maintain human health. Therefore, available drinking water must meet drinking water quality standard and doesn't pose any health risk over a lifetime. Consumption of drinking water contaminated is one of the biggest threats to public health. Globally, about 1 to 2 billion people are suffering from a lack of safe drinking water, and 30,000 people die every week from contaminated water. Water quality is generally assessed by several aspects that affect its quality, including microbiological, physical, chemical, and radiological characteristics, and the effect of these characteristics on public health. Most of the serious problems in drinking water, especially in developing countries, are mainly due to microbiological and chemical contamination. And microbial quality is the most important, especially pathogenic parasitic contaminants. Egypt is one of the middle-income countries that aim to improve the water supply to households. Government officials have blamed numerous outbreaks of diarrhea in recent years on water that was contaminated by poor pipe quality, as shown by the high bacterial counts at the water use point compared to the treated sites. However, a scant study on drinking water contamination is a concern because of the poor quality of the pipes connecting households to water treatment facilities in Egypt. Studies show that improvement in water quality and increased water supply are effective in reducing morbidity caused by diarrhea. This has been confirmed by a number of studies in Egypt that access to better quality water is associated with lower child mortality. More than 3.4 million deaths around the world from waterborne diseases. Drinking water has a range of risks in the form of pathogenic parasites, and if these parasites are not properly disposed of, they can pose a threat to public health and environment. The highest priority and the goal that all countries seek with the World Health Organization by adopting standards and laws to keep water safe and clean to prevent the spread of the disease, as waterborne diseases are the leading cause of death worldwide. Waterborne diseases are the diseases that are transmitted to humans or animals through water, which usually contain a proportion of pathogenic microorganisms. In this presentation, I'll focus on infectious parasites that either have been directly linked to the drinking water supply or for which there is strong evidence that transmission can occur by waterborne route. Firstly, protozoa. Protozoa are free-living single-celled organisms that have the ability to reproduce in humans and can transmit it from one human to another through fecal oral routes, through contaminated water, food, person-to-person contact. The second one is the helminthes. Helminthes are multicellular organisms that may be free-living or parasitic, and there are three groups of them. Flatworms, which include nematodes and cystodes, nematodes, which are roundworms and flat, and acanthocephalon, which are intermediate between cystodes and nematodes. Several species of protozoan pathogens may be transmitted to humans through the drinking water route associated with many health risks. The following protozoa and worms transmitted by drinking water according to WHO, as we see Acanthamoeba calpersoni and Cryptosporidium parvum, Cyclospora, Entamoeba histolytica, and Giardia. All are highly resistant to permissible chlorine and highly health significance and highly relative infectivity. Although Naegleria fowleri has low resistance to chlorine, it has highly health significance. This table showed parasite transmission through drinking water, but for which evidence is inconclusive or lacking, like Plantidium coli, Blastocyst, Isospora, and Toxoplasma, and also all have highly resistant to chlorine. And other examples for helminthes like Fasciola, free-living nematodes, and Schistosoma. In Egypt, studies have confirmed the presence and isolation of many pathogenic parasites transmitted through drinking water. The following table showed waterborne parasites during the last 5 years from 2015 to 2019, and these parasites are very similar to those table approved by WHO. Now I highlight for the most prevalent parasites globally. There are two parasites of free-living amoeba have been responsible for serious disease. The first one is the Naegleria fowleri. Cerebral infection by Naegleria fowleri is strictly waterborne, and although rare, is usually fatal. It's called the brain-eating amoeba. Enter the brain through nose and consume its cells. When people swim in warm, fresh water during the summer, people inhale through their nose, naturally making them reach the brain directly. These amoebas cause headache, stiff neck, and vomiting. These symptoms develop dangerously as the amoeba develops inside the brain and eventually reaches coma and then death. The second one is the Acantha amoeba. It's a microscopic amoeba widespread in the surrounding environment. It's found in nature and treated water in ponds and heated baths, soil, air, in cooling towers or heating, adaptation, and ventilation systems. Most people will be exposed at some stages to this amoeba without any medical condition. But Acantha amoeba is capable of causing different infections in humans. Keratitis is an eye disease that usually affects healthy people and leads to blindness. Granulomatous epiencephalitis is a serious disease of the brain and spinal cord, usually in people with a weakened immune system. Symptoms of granulomatous epiencephalitis like headache, stiff neck, or head and neck pain, sensitivity to light, nausea, vomiting, low temperature fever, weight loss, and difficulties in pronunciation and walking. There are two enteric protozoa causing severe disruption are Cryptosporidium and Giardia. Cryptosporidiasis is a diarrheal disease that causes by microscopic parasite called Cryptosporidium. The parasite is protected by an outer shell that allows it to survive outside the body for a long period of time and make it very tolerant to chlorine disinfection. Cryptosporidium is the leading cause of waterborne disease among humans in the United States. Symptoms of crypto begin 2 to 10 days after infection. The most common symptoms are watery diarrhea, also stomach pain, dehydration, nausea, vomiting, fever, and weight loss. The second one is the Giardia. Giardiasis is one of the most common diseases transmitted by contaminated water, causing chronic diarrhea and malabsorption that make the patient in a deplorable state. According to WHO, diarrhea is the leading cause of death among children under the age of 5. And Giardia is a widespread parasitic infection and the most prevalent in the world. 15 to 30% of children in developing countries under the age of 10 suffer from giardiasis. Infection with the parasite occurs through drinking contaminated water. The majority of people with giardiasis don't have any symptoms. The rest of them have giardiasis with a bundle of symptoms that vary from person to person, like diarrhea with the fuel odor, excessive gases, upper abdominal pain, and lack of appetite. And now I'll talk about international standard of drinking water. Guidelines for drinking water quality were issued in 1963 by WHO, followed by several guidelines for drinking water quality and development. In 1974, the United States established the U.S. Environmental Protection Agency, which is responsible for setting drinking water standard as a result of growing concern about waterborne diseases. As we see, WHO recommended guidelines for parasites starting in 1971 with the development of the following editions. The WHO established guidelines for the parasites. This edition considered many recommendations according to drinking water safety, including parasites hazards, which continue to be the important concern in both developing and developed countries, as we see in this table. Egyptian standard for drinking water. The Egyptian standard achieved the following objectives: make the water clear, free from color, potable taste, and smell, disposal of salts and harmful chemicals that limit the use of this water in human and industrial purposes, such as iron, manganese, water hardness, and etc. In this regard, on 1995, for the first time, decree of the Minister of Health and Population, number 108, regarding the standard specifications for drinking water, which is divided into 5 criteria. Physical properties like color, taste, odor, turbidity, pH, inorganic chemicals, which include dissolved salts, iron, manganese, water, and chemicals, which include inorganic and organic substances. Inorganic substances like heavy metals, lead, cadmium, and organic materials such as pesticides. Microbiological criteria include total number of bacteria, indicators for contamination, total coliform bacteria, colon fecal bacteria, and streptococcus. Biological examination of protozoa and blue-green algae and radioactive material like alpha and beta family. Egyptian standard specification indicates that drinking water should be available and should be free from protozoa and all stages of pathogenic worms. A new decree in 2007 was issued by the Ministry of Health and Population on the maximum standard specification to be met in drinking water and domestic use. With regard to biological test for parasites, there was no change from the previous one. And it is the same also for potable bottled natural mineral water and bottled packaged drinking water other than natural mineral waters. Conclusion. Access to safe drinking water is a fundamental human right to maintain his life and public health. In order to ensure access to safe drinking water, the government issued the standard on drinking water quality based on the World Health Organization drinking water quality guidelines. Regarding microbiological standard in Egypt, the standard don't require specific parasite screening as in the United States of America, which the US EPA require Cryptosporidium and Giardia lambelia tests. In addition, the US EPA recommended removing 99.9% of Cryptosporidium and Giardia lambelia before water consumption. Recommendation. Monitoring of drinking water quality for contaminants from the responsible authorities. Development of drinking water quality standard and the addition of any possible pollutant can be detected. Ongoing maintenance and the need for continuous replacement of water supply and sanitation systems. Protecting and conserving different water sources from pollution and with the appropriate treatments. Health education for the community about pollutants and the importance of access to safe water. Interest in scientific research in the field of developing new technology for studying waterborne disease. Thank you for your attention.
Thank you very much, Dr. Wafa. Do we have any queries? Okay. Dr. Anahit, are you available now? And may I know Dr. Anton Holland and Ricky Bates available for their presentations? Okay, we'll take a short break of 5 minutes and we'll be back with keynote sessions.
So, the- we are left with 3 presentations right now. So as we are not able to contact Dr. Anton and Dr. Ricky, so as being Canada and USA, maybe they might need some time. So let's just go for a 40 minutes break, and in the meantime, we'll just try to contact them and meet back again at 40 minutes later so that we'll just have the other presentations going on. Thank you. For any queries, please do contact us.
Yeah, hello everyone. So we will have the keynote presentation now from Dr. Deepak Sandra from University of Nebraska Lincoln, Panhandle Research and Extension Center, USA. And the presentation title is also Millet as Climate Resilient Food Grains for Human Health. Dr. Deepak, are you ready to present your talk? Dr. Deepak, can you hear me? Dr. Anand, we can see your login, but you are not responding. Everyone is waiting for your talks. Please respond. Hello everyone, can you hear me?
Yes.
Yeah, so we'll have a keynote presentation from Dr. Anton Hall and Niva in Canada. And the presentation title is Changing the Channel on Communicating Climate Change to the Public.
Thank you very much.
Yeah, okay, I'm making you the presenter.
Okay, I don't have any slides, I'm just talking, so.
Yeah, okay.
Thank you. Thank you for your presentation. So, do we have any questions? No questions.
All right. All right. So I'll just get started.
Okay.
Oh, okay, okay. Yeah, please do go on.
Are we good to go? Is everything good to go?
Yeah, you can start.
Okay, great. All right, well, thank you, everyone. So public concern about the impacts of climate change is the highest it's ever been. But so too are our planet's temperatures and the level of carbon emissions that are driving them up. You know, warnings from scientists and environment agencies about what's happening to our climate aren't in short supply. Still, we've ended up at a point where now we have about 10 years to change course, to curb our carbon emissions enough to prevent the Earth's temperature from rising more than 1.5 Celsius above pre-industrial levels. And beyond that point, according to the UN Intergovernmental Panel on Climate Change, we'll see even more catastrophic impacts in the form of extreme temperatures, droughts, and more intense storms. The problem is that even people who express concern about our changing climate don't necessarily connect with messages like that, and they don't automatically feel compelled to change how they live and make sacrifices to lessen their own impact on our climate. Engaging people takes a more simple approach. We've been assuming that information transfer, knowledge, will help people. If we just give them the science, they will naturally be aware of the impacts and start changing their behavior. But reviews show that people can feel paralyzed by all of the information. They can't possibly take it all in, and then they just check out. And on top of that, we have the vocal pushback, the people accusing those of us who talk about climate change as lying about it, being part of a cult, or profiteering off of it in some way. And that kind of commentary can dominate the discussion, and it's confusing for a lot of people. But the reality is that those people are in the minority. They just do a really good job of getting their voices heard and getting their point across in a way that is compelling to people who may be confused by all of the science. In Canada, where I'm from, the reality is that nearly 80% of Canadians agree that the planet is warming, and nearly 2/3 of the country recognizes that the main reason for this warming is due to human activity. Our biggest challenge isn't skeptics who perpetuate the idea that science is somehow optional or a matter of opinion. It's that when it comes to supporting climate action, the urgency just isn't there for many of them. How many times have you heard people say when they're scraping the ice off their cars in the middle of January that they would like a little global warming? As for solutions, we wonder why we should bear the brunt of the financial impact, or what we can do that would actually have some kind of global effect. Remember when I said that climate change feels like a problem that is far off in time and space? The reality is that climate change is affecting us today, here and now. And it's doing this by taking many of the risks we already face naturally, things like floods, storms, heat, and drought. And it's supersizing them. It's making them significantly worse. A common misnomer that's talked about with the public is that we need to do something to save the planet. This isn't about saving the planet. The planet itself will survive just fine. The question is, what will happen to the rest of us who call the planet Earth home? Will we survive? And if we do, what will we be left with? So as scientists, that's exactly why talking about climate change is so important. If we don't talk about why it matters, why would we care about the problem itself? And if we don't talk about what we can do to fix it, why would we take action or expect our communities, our governments, our countries to do so either? As challenging, as stressful, and as difficult and frustrating as it might be, fixing the climate change problem begins by actually talking about it. It sounds easy, but it isn't. The key is to focus on why climate change matters to all of us. We can all point to instances in our own countries where climate change is already leading to things like more frequent heavy rain events and record-breaking heat waves. Catastrophic losses due to natural disasters have increased dramatically, with insurance claims rising just as rapidly, amounting into the billions every year. Along coastlines all around the world, rising sea levels are combined with stronger storm surges to threaten coastal communities and infrastructure. In the north, we have thawing permafrost, sea ice that develops later and disappears earlier every year. Inland, we have rampant wildfires in places like Canada and California. Farms are running out of water. So why, as scientists, or me as a science communicator, is it so difficult to get the message across to people? Basically, the problem is we haven't been able to get the average person to connect the dots between what's happening, why it's affecting them, and what they can do to be part of the solution. Like most of the world's problems, it comes down to understanding people and bringing the conversation down to the things that concern them the most, the things that they identify with the most, the things that affect them as people. And then, after that connection is made, there's plenty of room to slide in the science. Do they like to fish? Talk about warming oceans and why it's been harder to catch the things they've always been used to catching. Do they like to ski? Talk about why there may be fewer good ski days because it's been harder to hold on to skiable conditions for a decent length of time each year. Are they concerned about their grocery bills? Talk about why it's harder to grow some of the things that they like to eat, which results in higher costs at the checkout. Get to know them. See if there's some common ground on which to base a conversation rather than a one-way delivery of information. You're never going to win someone over by overwhelming them with data and facts and figures. And you can't meet on common ground if you go hard out of the gate with something the person you're talking with flat out rejects. But you can make some progress if you start by addressing their values, what makes them tick. Like any good conversation, you need to show respect for them and where they're coming from. Maybe it's a place of loss. Maybe it's confusion. Maybe it's even fear. Help them to connect the dots between what they care about and what's actually happening out in the world. And then provide them with the means to see a positive outcome, because that's what actually motivates people to act. For example, a big part of the conversation is how we need to limit or eliminate our use of fossil fuels to reduce emissions and our carbon footprint. But tell that to someone from an area that traditionally relies on the oil and gas industry, or someone who directly works for it, and you can see where this is going to go. You're directly attacking their ability to make a living, to pay their mortgages, to raise their children, to just do the things that everyone should have the ability to do. But if you can genuinely show that those things matter to you because you are concerned for them as a person, and then show that a transition to clean energy actually brings with it new technology, new opportunities, new investment, more money for them and their families, as well as a brighter future without all of the uncertainty that the effects of climate change can bring, then suddenly you will have completely changed the channel on that conversation. You'll no longer be adversaries, but two people trying to sort out a common problem. Thank you.
Thank you very much, Doctor. And can we see your slides? Because we cannot see your slides while you're delivering your talk.
I did not have any slides.
Okay, that's fine. Okay. So do we have any queries? Okay, so next keynote presentation by Dr. Ricky M. Bates from Penn State University, USA. So presentation title is Use of Perennial Wild Food Plants to Diversify Cambodian Agriculture for Improved Nutrition and Income. Dr. Bates, can you hear me? Hello, Dr. Ricky M. Bates, can you hear me? We are waiting for your talk. Hello, Dr. Ricky, we are waiting for your presentation. Can you please respond?
Dr. Ricky
Can
Bates,
you hear me now?
yeah, now we can
Can you
hear
hear me
you. Yeah.
now? I'm having some technical difficulty on this end.
Oh, but now your voice is actually clear. It's visible. It's audible, sorry.
Okay, can you see my screen yet?
Oh, not yet. We're just waiting for it.
How about now?
Oh, not yet. Like, have you clicked on the screen button?
All right, let me try that.
Mm-hmm. Oh,
I click on the screen, but nothing seems to be happening.
I have already made you the presenter. Yeah, it's actually you are the presenter right now. So you can just click on the screen. So a pop-up window will open, or maybe you haven't given any permissions. Is that so?
It says open system preferences.
Oh yeah, so it's actually the settings issue. You must give the permissions for GoToMeetings to access your system.
Okay, I'm in GoToMeeting.
Mm-hmm.
And I'm in settings, but I don't see any
So in any case, could you send us an email with your presentation so that I can present it for you and you can just give a talk?
Okay. And this
Yeah.
would be to
So
Sharon Charles,
oh yeah,
correct?
that's fine. You can send it. Yeah, correct. You can send it.
Okay.
Yeah.
Okay, I just sent it. Meanwhile, I will continue to look for a way to.
Yeah, you can share the PPT at my email, sherincharles. So that I can
Yeah,
share your
it's
PPT.
yeah. It's sending right now.
Okay. Hello, Dr. Ricky. Have you shared the PPT?
Okay, I have shared my PowerPoint presentation. I've
Okay.
emailed it, but I tried to restart GoMeeting and it's showing that it's not letting me share my screen.
Okay, it is allowing or not?
Pardon me, I'm online, yes.
Yeah, you can share your screen or
Yeah, it's not letting me share my screen.
Yeah,
Let me try again.
sure, you can actually try again. Dr. Rick, your screen is visible to us. So yeah.
Up. Okay. Can you see it now? I think I figured it out. I had to go through
Oh,
several
yeah, it's
steps here to
perfect
get
now.
that to work.
It's perfect.
Okay. Okay, great. My apologies for that.
No problem, no
Okay.
issues.
Let me just minimize this here.
Oh yeah, for that you'll have two arrows facing the opposite side. So just click on that. So it'll just get minimized.
Okay, there, I got it out of the way.
Yeah.
Okay. Okay. Alrighty. Sorry for the delay again. I'll try to keep this to 15 or 20 minutes. Sharon, if you just let me know when we're running out of time or
Oh yeah,
or
no,
when I
no
have a.
problem. You can actually complete the talk without any issue. No problem.
Okay, great. So
Mhm.
this talk deals with a technology that is related to sustainable intensification called wild gardens. And it's a project that we're working on in northwestern Cambodia with colleagues at the University of Tennessee, as well as the National University of Battambang. And this is part of a larger USAID funded project called Women in Agriculture Network Cambodia, that looks at the intersection of sustainable intensification practices and gender. As the horticulturist at Penn State University on the project, my role in the project dealt more with the technologies and less on the social science side of the project. And so we had as an objective 3 areas of focus. One was post-rice cover crops and relay crop systems. We were also looking at rainy season vegetable production, primarily looking at the technology of vegetable grafting. And then we conducted proof of concept research to characterize what we're calling wild gardens. And so when we're referring to wild gardens, we're really talking about neglected and underutilized species. These are sometimes called orphan crops, or more recently, wild food plants. And these are plants that have had little research done on them. They're usually not in the commercial formal seed system. They're more locally adapted species. Some are indigenous. They may be semi-domesticated. So we concentrated on these wild varieties of trees, shrubs, vines, and ground covers that some of the population actually use for food. And these are underexploited species that have potential for improved health. Some of them are medicinal plants, improved income. They're generally present in the marketplace, and certainly environmental services in terms of things like carbon sequestration. So they do have a role relative to climate change. For instance, some areas maybe are annually row cropped that should be put in permanent species, and these could offer an opportunity there. And so when we're referring to wild gardens, we really mean a strategic grouping of these low maintenance, often perennial wild food plants. And we wanted to optimize and promote this as a tool for sustainable intensification on underutilized land. These would be places like field margins, vacant plots, farmholder homesteads, where they may interface with a more traditional vegetable garden, including the rice paddy. And so any village in Cambodia has a lot of this type of land, as you can see in the picture on the left. Excellent functioning wild garden, as shown on the right, often just looks very jungly and chaotic. But in fact, the mix of species that are there are strategic, they're thought out. Most of them are multifunctional. And so we went about characterizing this system in 2 provinces in Cambodia, in Battambang province and Siem Reap. And we had as partners World Vegetable Center, National University of Battambang, Eco Asia, which is an NGO operating in Southeast Asia, University of Tennessee, and then my home university, Penn State. And we went about this research through key informant interviews, gender studies, farm analysis, farmer interviews, market surveys, a whole mix of different approaches to characterize the system. We developed market and farm wild food plant inventories, collected a number of different demographics to just characterize the presence and the place and the usefulness of these species in the food system in northwest Cambodia. So there are a number of food security issues in Cambodia generally, widespread land degradation, especially rice land. Yeah, most of the country relies on rain-fed single harvest crops of rice, meaning that these lands then often lie fallow for much of the year. Household diet diversity is low. There are migratory issues, young men leaving rural areas for urban centers for work. And so diversification of smallholder farms is an important step in addressing some of these food security issues. The value of a wild garden can be that it can diversify these smallholder systems in a temporal, spatial, and functional diversity. And so there's a lot of information on this slide, but what I want to point out is that usually in Cambodia, in rural small farms, there are usually 2 what we would call food gaps, times of the year when it's difficult to grow traditional annual vegetable crops such as tomatoes, eggplant, chilies, those types of species that are normally associated with an annual vegetable garden. There's a hot, dry season, and then there is a wet season. And so the wild food plants assembled in these wild gardens that we've prioritized can function and produce during these 2 food gaps. So in addition to the temporal diversification, there is a spatial diversification that's possible in the tropics with these species. And so our perennial wild food plants, when assembled and selected correctly, can occupy all 4 layers of these niches. And we're relying here on trees, shrubs, vines, and ground covers. But here's just a schematic of some of the species that could be used that basically fill those 4 layers, adding the spatial diversification. And then many of these species, such as moringa, lucena, galangal, chaya, several bamboos, sun hemp, are multifunctional, meaning that some of them can be used in addition to human food, livestock, fodder, living fences, income from local markets, and even energy sources because of the oil content of the seeds. And so one of the areas we focused on was nutrition. And so many of these wild food species like moringa, chaya, acacia are nutrient dense. And so using these species and integrating them into the household food system or the community food system is a good way to diversify diets and improve the dietary quality. They're also low input and low cost. I would just point out as an example, Acacia pinnata. Up until this project, no good analysis of that plant, nutritional analysis had been conducted. And so one of the things that we did with some of these underutilized species was conduct nutrient analysis. And in the case of Acacia pinnata, it's quite high in beta carotene, the precursor to vitamin A, which is a problem. Vitamin A deficiency, childhood blindness is a problem in many Asian countries, certainly in Cambodia. And by incorporating this single species into a wild garden, can address that particular issue. And then the other thing that we looked at closely was income. We did extensive market surveys and worked with farmers to characterize how they're using these products in the market. And this is important because for farmers to, I think, sustainably grow these plants and for them to be important on a local level, there needs to be market pull. And many of these species are present in the local market. Some of them have very good income potential, particularly at times of the year when the farmers aren't getting money from other more traditional species. We surveyed just under 75 farmers who had assembled and were using these wild gardens to some extent on a continuum, and asked them basically what are the benefits of maintaining a wild garden. And you can see almost all the respondents at the bottom bar consume these plants at home. Almost all of them, 95%, were selling these plants in some way or making money from them or trading them with their neighbors. They didn't often realize the nutritional benefits of the plants. And so this survey also revealed some areas where education is necessary so that farmers can understand the full range of benefits. For instance, not many of the farmers were thinking about soil quality, where in fact, we've done some preliminary research showing that these perennial species, especially over the course of just a couple of years, can improve soil macro and microfauna. They also replace expenditures on household expenditures on food, medicine, pesticides. And again, using Acacia pinnata Saom as a case study, this was a cash crop present in the market, and so it was an important source of income. There were some women farmers who had formed a cooperative under our guidance in this project to form a nursery business around this particular species. High nutrition, especially vitamin A, and it's used often, it's a shrub, but it's used often as a living fence to control livestock. And so the plant will be trained in a way that makes it kind of an impenetrable fence. It has a thorny stem. And so this was a very good multi-use plant that we saw particularly in Battambang province. For our monitoring and evaluation, we looked at aspects of productivity, economic, environmental, human, and social realms. So we found that women are generally the wild garden caretakers. They don't have a lot of time to manage new enterprises, and so these plants work well because they're low maintenance after they're established, unlike a traditional vegetable garden. We also found that household nutrition and biodiversity potentially improve, but they're not often felt needs. They don't often understand the nutrition present, and sometimes even with some species, we don't know what the nutritional component of the plant is. And then also there were things like important community building functions, because these are informal plants in the informal seed system. You don't go out to a nursery and buy a lot of these species. They're traded between family members, neighbors within a community. And so it's very complex because it's part of the informal local seed system. And so we felt like promoting and working with this particular aspect of the food system was important. We felt it was a very suitable, sustainable intensification technology for Cambodia, but asked the question, is this something that's scalable? And if we do attempt to scale, how sustainable is that? What are the constraints? There are opportunities here. This mimics a natural ecosystem. and provides a high level of sustainability once established, and it's resilient because it's adding diversity and also serving as a low input food system. It complements home vegetable gardens, but it's less resource intensive. This is important because farmers who don't have vegetable gardens and have low dietary diversity are often adverse to adopting more traditional vegetable garden approaches. And so this represents low farmer risk, but it could also be used as a standalone technology. There certainly is documented income from market pull. And the other important regional aspect is that it's insulated from regional import competition, primarily from Vietnam and Thailand, where a lot of vegetables are imported. And so a lot of these species are unique from a culinary standpoint in the Cambodian cuisine. And then finally, we feel like this is a very gender sensitive approach for supplementing income and nutrition and addresses this feminization of agriculture based on outmigration of a lot of the labor force, particularly young men. There are some constraints, so ramping up or scaling germplasm is one potential problem because it is part of the informal seed system. It's a traditional system, but a lot of the indigenous knowledge is eroding. A lot of the know-how, for instance, is not being passed down from one generation to the next. It's not part generally of the Cambodian agriculture education and training curriculum in the schools. And then again, the outmigration from rural smallholder farms creates a lot of risk adversity. And so that does need to be dealt with. And with that, I'll take questions if there's time.
Thanks a lot, Dr. Rick, for your presentation. So, yeah, do we have any questions? Any questions to Dr. Rick about the presentation or any comments or anything that you'd like to share? I guess there are no questions. Thanks a lot, Doctor. So let me just help you. Yeah. So, uh, oh yeah. Thank you, Dr. Rafa Hickel. So I guess we'll be pending with one more presentation by Dr. Deepak Sandra. I completely understand the thing, but I'm really sorry for making you all wait. But we have already been in contact with Dr. Deepak, and as it's early in the morning for him, he'll be just joining us in 5 minutes. And I request you all to please be patient and waiting. And Dr. Rick, I'm also thankful for you to joining us at a very odd time and very early in the morning for us. Thank you. Thanks a lot for your patience, and yeah, we really appreciate your efforts in this. Thank you.
Oh, you're more than welcome.
And
I had my time zone. I thought that I was on at 9 o'clock instead of 8 o'clock. So I'm glad I had tuned in as soon as I was up and online, and so that was good.
Yeah. And also Dr. Anton Holland. And yeah, I'm really sorry. I guess I've disturbed you a lot by all the calls and early in the morning for you. I'm so sorry for all that thing. But yeah, as we had a lot of Asian participants today, like for this program, so considering their timing, so we had to organize the webinar at a British time. So that has caused all this. So yeah, I'm so sorry for this. And I really hope you guys wait for just 5 minutes so that we can have the presentation of our next speaker, Dr. Deepak. Thank you. Thanks a lot for your patience. and we request you to take care of all these time differences for the next time for sure. So yeah, this will be the time conducting a event like this. But yeah, for the next time, we'll definitely follow the Eastern time for the webinar so that it'll be helpful for every person attending the conference or the webinar. So let's wait for Dr. Deepak just to join the webinar. I just now had a conversation with Dr. Deepak, and he said that due to some internet connection technical issue, so he just said that he's driving off to his office right now. So he might take another 10 minutes to reach the office and to join us. So I'm not sure if it's good that I'll make you all wait for such a long time. But yeah, it's important that we also have to listen to Dr. Deepak's presentation. So I'd like to inform you that I really hope everyone can stay up till that time. And in the meantime, I actually have one more presentation left by Dr. Uh, just give me a second. By Dr. Anahit Alexandrian from the Ministry of Environment of the Republic of Armenia. So I just want to see if Dr. Anahit is available online because I could see you attending the webinar, but I'm sorry I couldn't make you get the presentation. Like, I'm not sure if you're online or just logged in or anything. So can you please let me know if you're online so that we might have your presentation now? Okay, no answer. So anyways, so as I've mentioned, it might take another 10 minutes for Dr. Deepak to join us. So in the meantime, if anyone is interested, please be hold right here. And for others, I do understand your busy schedule right now. So we really hope you, if you can stay back till then. But yeah, if you are having any important or any busy schedules, yeah, we'll definitely share the recordings at YouTube, and we'll share the links to you all so that you can once again recap all the presentations online. So yeah, it might take next 10 minutes for Dr. Deepak to join. Thank you for your patience.
And
Yeah.
so
Hello,
with
Dr. Deepak.
we actually come to the end of today's session and also the webinar. And yeah, so I really thank each and every one of you for your patience and all of your presentations. It really like I hope everyone has gained some knowledge about each other and all of your presentations. And thanks a lot. Regarding the certificate will be sent to you through email after the webinar is accomplished. So once the
Hello, Dr. Deepak. Can you hear me?
Yes,
Yeah. Are you ready now?
yes, I am ready. I apologize for this mess. I'm sorry, but here I am.
That's fine. Next time we will organize the webinar in Eastern timings for sure. This time we have almost 10 to 12 presentations from Asia Pacific region.
Yeah,
So
I realized
if we
that.
organize any,
Yes, yes.
yeah, yeah, but few speakers are questioning about this thing. But next time we'll take care of that and we won't make it a mess.
Oh, sure, sure.
Do you have the slides or you'll present it directly?
I have opened slide. I can share.
Oh, yeah. Okay, you can share now. We have given you the access. Yeah, we can see that.
You can see that.
Yes, yes, we can see that.
I'm trying to see bigger screen if I can make here, but looks like, let's see. Huh,
It's clear.
it's clear at your end?
Yes, it's clear.
Okay, all right. So, tell me who are the attendees, like which part of the world they are, just briefly, so that I know how to direct my talk. Okay.
Actually, we have the speakers from Australia, South Korea, China, Canada, Taiwan, Spain, Slovenia, Poland, Lithuania, and India, Turkey, Tunisia, Armenia, Saudi Arabia.
All right, sounds good. So, should I start?
Okay. Yes, yes, you can start.
All right. I guess good morning, good afternoon, or good evening to all over the world, whoever is attending. I'm Dr. Deepak Santra. I am attending from USA, and my local time is 6:52 a.m. and this is a local time, Denver time zone. That's what I am in. So I'll be talking today about proso millet. Okay. So the title I have given, the proso millet is a climate resilient food grains for human health. And in future, if you have any question related to proso millet or anything what I'm presenting here, you see my email address. You can note it down and you can reach out to me. I'll be glad to answer any of your questions. Come on. Why it is not moving to next slide? Okay. Okay, got it. Okay. Here's the proso millet. It's a Panica miliaceum. It's basically cereal, a grass family. It's a small grain cereals. From distance, it looks like paddy. And this is the proso millet field from my area here, from my research farm. I just wanted to show you how the proso millet looks like. The millet, the proso millet, there is no global statistics about its production for proso millet as such, but there are several millets. There are 7 different millets, which proso millet is one of them. So in the FAO, they have grouped together all the millets and put the statistics. So that according to that, India is the #1, Nigeria, Niger, China, and you can see that on the graph here, pie chart as well as bar graph, the top 10 countries for millet production. Now I'll remind that all the attendees, this millet is not for proso millet. This is a group of millet, which is the pearl millet, finger millet, proso millet, foxtail millet, barnyard millet, kodo millet. So these are the all different millets together. That's the global scenario. Here I am. I am located in the central Midwest of the US, western part of the state Nebraska, as you see here. I kind of marked it. This is the location I am in. So this area is the US proso millet growing area, and many times they call it as simply millet. The characteristics of this area, why proso millet here, why not corn, why not soybean, or why not apple, if you may think. This is a semi-arid high plains environment. It's a essentially high altitude desert characterized by short growing season, is limited rainfall. Annual precipitation is about 40 centimeter per year, including summer rainfall and winter snowfall. Temperature fluctuation is a huge temperature fluctuation. Sometimes daytime temperature could be about 35, 36 degrees Celsius, and night temperature could be 16 degrees. That type of temperature swing. Hail is a regular phenomenon here. I lose my breeding trial once in 3 years. And here is the moisture, water stress. So the red graph, as you see here, is actual reality, and this is the demand, very high ET region. So very drought prone area, limited soil moisture situation, short growing season, and high altitude. So that's the environment we are talking about. So my job is to develop a new crops. Proso millet is one of these. And as you see, short growing season here, we have to fit whatever crops we grow, we have to fit within the small window. Why we are talking about proso millet in US, or what is the justification of the alternative crops? Now this area is a dry land, winter wheat based dry land cropping system area. Traditional practice is a winter wheat and then fallow. What it means, like if you see my picture on my right side on the screen, this is called steep farming system. This is the current season. So this year they were growing just harvested the wheat, and the strip next to that is they will be planting wheat this year. So basically they alternate these strips for farming wheat. So it is a 2-year crop rotation from the whole plant. Like these strips will be fallowed next year. So you are getting one crop once in 2 years. So it is a 2 years crop rotation. And that means we are living about 14 months fallow period. So proso millet fits in that area, like this table here briefly. This is the traditional year 1 wheat fallow. But with the modern, for example, millet and sunflower, you get 2 crops in 3 years. Or if you put a 3 years crop rotation, wheat, millet, sunflower, 3 crops in 4 years. So I do not have to tell what is that implication of the leaving the land fallow. It's not good for the environment. It's not good to farmers. So we are increasing cropping intensity. In other words, potentially increasing farmers' profit margin, inserting some new crop like millet in the existing cropping system. So that's the place for millet. So I work on multiple different crops, cereals, one ancient grains, and then oilseed crops, and the legumes, and some other crops like amaranth, buckwheat, that kind of crop. So I work on multiple crops, but today's speech I will talk mostly about the proso millet and my program, what I'm doing, and what its relevance with the current climate change and human food security. What I was telling about millet is a group of annual grass or cereals, primarily originated from Africa and Asia. And here's the seed compared to, say, wheat, corn, barley, paddy. They are all cereals, but they are coarse grain cereal, is a bigger size. But millet, as you see, all the millets are displayed on the picture on my right side. There is very tiny grain. So that's characteristics of millet. Overall characteristics with respect to its relevance to today's world is a healthy to environment, is a healthy to human health as well. Is a gluten-free, high fiber, low glycemic index, high in minerals and complex metabolic diseases it helps. Why I'm talking about it is human climate healthy. Who does not know the problem with fresh water for agricultural production system? Water is becoming a major constraint for mankind, I guess, in future. Proso millet is human environment healthy. Why? It is shallow rooted, low input crop. It doesn't take much fertilizer, doesn't take much water. I'll get into the point. Adapted to semi-arid environment where major crops like paddy, wheat will grow, corn, soybean, barley. When this will not grow, proso millet can grow. Very short duration and can use the water most efficiently among all the cereals. See the bar diagram below. What this is, okay, it is cut off. Okay, I can tell you that from left to right, the graphs are basically this graph says, excuse me, amount of grain in ounces produced per gallon of water. So y-axis is that weight of the grain, x-axis is the crops. So first one is wheat. So how much grain is produced of wheat per gallon of water. Next one is barley, little higher. Maize or corn. Next one is sorghum, which is kind of millet category. And second last is foxtail millet. And the last one is proso millet. So it's clearly the proso millet is the leader in water use efficiency among these 6 cereals this graph, this study presented. So proso millet is the highest water use efficient cereals so far known. Here is that evidence. In 2012, we have historical drought. Here is the corn field, and this is the millet field. These two fields were about a mile apart. What the precipitation was very low in that area. See the situation of the corn. There will not be any single kernel to be harvested on this field. Whereas the proso millet, I will get at least about 1,000 pounds or 1,500 pounds per acre proso millet here. So that's the beauty of proso millet with respect to climate situation and water limited condition. Now I'm talking about proso millet industry in US, how big it is. Of course, it is not 10 million acres in US. Total average production is about 500,000 to 750,000 acre in the USA, and total value is about $26 million to close to $80 million. And as I indicated this by this map, these are the 3 states which produces about 95% of total millet in US. Colorado is the leader, which produces about 50%. Nebraska is another 25%. Then South Dakota here, this part is probably about 20%, and remaining 5% elsewhere, like this part of Wyoming, North Dakota, Kansas, little bit, Minnesota. So these are the proso millet growing area. My program here in the University of Nebraska is the only proso millet breeding program in the Western Hemisphere. And this program started in 1965. And this is a historical information about the proso millet breeding program past and future. The first variety was developed in 1967 called Panhandle, and then there are a series of varieties, and then I started working on that on the different breeding tools. I'm a breeder and geneticist by training. So whatever proso millet varieties are grown today in US or North America as a whole, they are primarily coming from this breeding program. Breeding targets, of course, high yield. Then agronomic traits, early maturity, obviously because of short growing season. Otherwise, my grains will be frosted. Uniform maturity for direct harvesting, lodging and seed shattering resistance. Because of this inherent nature, proso millet is lodging susceptible as well as shattering susceptible. So farmers lose grains, lots of grains on the field if they do not harvest on time. And this area is very high wind area. When proso millet is mature, our wind speed could go to 30-40 miles per hour. With a dry is very favorable situation for losing grain from the proso millet. And specialty traits, focusing on human food and other industrial uses. It's very standard breeding panel. Is a self-pollinated crop. So you just make cross, generate the hybrid, and they advance the population. As you are advancing, you are testing in different design to identify the high yielding, most adapted variety. At the last stage, you test those handful of lines under multiple locations to release the variety. And it's just crossing picture here in the field as well as the greenhouse. And then obviously, because this is the area where proso millet growing area, so obviously my multi-location trial is all over this world, all over this Colorado and Nebraska, of course, and South Dakota. Now, a little bit about my research. As a breeder, obviously, I need to test for my germplasm pool, whether I have the right type of characteristics I'm looking for, which I can incorporate in my to be released varieties. So we have the world germplasm collection. We have characterized that. This map just shows where the germplasm came from, primarily Asia. So those are the germplasm came from. We did test in the field to identify the most useful lines. As you see, these are the important traits I summarized on this table. The left column is the different characteristics like heading date that kind of dictate you that whether it's early maturing or late maturing, all these things, shade shattering, lodging, head type. And as you see here, so basically what we have identified, some of the tolerant to moderate tolerant line for my desired traits, so that I can utilize those in the breeding program in the crossing panel. So these are just sharing few of the research, and here is the cluster analysis. So what it helps, all these lines are clustered into 76 different groups. What it means, like obviously by breeder, if any of you are breeder or geneticist, in order to get the maximum diversity, you need to select one parent from this group, for example, another parent from this group, to capture the maximum diversity in the progeny. Those are based on morphological marker, and this cluster analysis is based on molecular marker, similar kind of situation. What it helps, although morphologically they may look similar, but based on DNA marker, they may look different. That's the reason we had to do that, and then select the most diverse parents. Here is that molecular linkage map. Proso millet, unlike corn, soybean, wheat, rice, tomato, and so on, there was no genetic linkage map developed before me. So we were able to develop a preliminary genetic linkage map for identifying some gene, for mapping some gene. Here are some of the QTLs, what we identified, like grains per panicle, like which is one of the yield component, lodging, some of the grain shattering, plant height. We have identified those QTLs, we mapped those, and we are refining those maps to identify better markers so that we can utilize genomic selection in our breeding panel. This table briefly tell you all these characters. This picture I just illustrated just 4 important QTLs here, and these tables identified all the 19 QTLs for 8 different traits what we have identified. So traits are on the right left column, and then the QTL's name, and essentially these numbers, Lord, and that phenotype variance explain that numbers kind of tell you how strong those QTLs are. But by default, anything above probably 3 Lord value is good, and obviously higher the number of this value is better. For example, internet length. This QTL 35%. So this is a major QTL, although Lord value is little lower. Now, so this is briefly I told about my proso millet breeding, what I'm doing, and I introduced you what's proso millet, and now I'm talking about proso millet with relevance to human food. Traditionally in US, proso millet today is predominantly utilized as animal feed, as a livestock feed, bird, hogs, cattle. But globally, millet in general, including proso millet, is primarily used for human food. The different types of food like steamed bread, puris, pasta, and industrial use like, of course, alcoholic beverage. As you see on my right panel, this is that popular, probably 60% of US millet is used for bird feed, but worldwide, predominantly it is for unique foods and beverages. That's the way proso millet is being used. Why? Proso millet is not junk. Just briefly sharing some information, nutritional information. These are the few of the varieties what our program developed, and this is the major nutrients in the seed composition. So starch is about 65%, protein not bad, about 10, 11, 12%, then fiber, fat, and ash. What it tells me, I'll come to the next point, proso millet seed composition is very comparable to other major cereals with respect to human food. See that this is the nutritional composition of millets and other cereals. If you see that my left column has all the cereals: wheat, rice, maize, sorghum, barley, oats. Proso millet is here, which is bold here. If you see the protein, for example, okay, wheat, so these data, wheat has about 14%, but there are other cereals which has the protein, and proso millet is 11%, not bad. Fat 3.5, crude fiber 9%, really higher than many other cereals. So take home message, proso millet seed composition is very similar to all other major cereals we are familiar with. Now, what about minerals? Similar situation, although some minerals probably a little bit higher compared to others. And these are again same thing: wheat, rice, maize, sorghum, and here is the proso millet. If you compare the numbers in across that different rows, you will see they are very comparable here. Like this is calcium, phosphorus, potassium, sodium, magnesium, iron is really high in proso millet. Manganese, zinc, and some of the vitamins. So proso millet nutritional profile is similar to other major cereals. So there is no reason to exclude proso millet from human food. Okay, and because of that, we started working little bit of proso millets towards developing American market compatible food products. So one of the research I'm just sharing, we developed a proso millet extrusion method. What extrusion is, I am sure all of us we know instant oatmeal cereals. So before you can eat as an instant oatmeal cereal, that raw oatmeal needs to go through the process called extrusion, so that is instantly cooked. And that's the proso millet. There was very limited or no report, so we developed that one. And here is the comparison. As you see that the extrusion method for all other grains, here are the characteristics, and here is the proso millet. So it's very similar. We established that. That means this research data can be utilized for developing instant proso millet based breakfast cereals. And in fact, they are already in the market. One of the issues with proso millet we identified, unlike many other millet, proso millet protein digestibility, although protein is about 12% cereal protein, but its digestibility, its availability to the human nutritional system, like human physiology, is not that good. As you see here, here is the proso millet total protein, okay? And this is uncooked, and this is after cooked millet flour, the protein in the millet flour. And when we isolated that, same thing, this is the total protein and after cooking. So there is a significant difference after cooking, how much protein will be digested. And primarily because of is a prolamine fraction of protein is a panisin. That protein is the predominant protein. That protein is not, it is easily accessible to the humans, the digestive system or digestive enzyme. That is the primary reason. So only way to improve the protein digestibility is to identify genetically superior genotypes using the crossing program. That is one solution. Other solution, do little bit characteristics of those protein fraction, identify those problem amino acid residues, and maybe improve that protein digestibility using gene editing technology. Once you identify the gene, prosomillet genome has been sequenced, protein genes has been sequenced, so there is a chance we can do that. Next one is a prosomillet for alcohol production. This study we did that compared with the high fermentation corn. As you see here, prosomillet, this is the starch, okay? This dotted line, this is corn, open box, and this bottom line, which is the closed square, this line is the prosomillet. So as you see, this y-axis is the ethanol concentration and x-axis is the fermentation time. So as fermentation time is going up, the starch is decreasing because it is being fermented. Very similar kinetics between the corn and soybean, and at the same time ethanol production is increasing. So take home message here, we know corn has been used for industrial scale ethanol production. We wanted to compare whether prosomillet stands there. Is it comparable to corn? And the answer is yes, it is comparable to corn with respect to starch fermentation as well as ethanol production. So the starch quality what prosomillet grain has is highly very similar fermentable level like corn. Gluten-free noodle from prosomillet. We set an experiment. Here is the uncooked noodles based on wheat flour, and here's the cooked based on wheat flour. Here is the prosomillet. So prosomillet flour can be utilized to produce gluten-free noodle. There are few slides I'm going to present about the prosomillet for human from some parts of the world. This is the prosomillet like what you can buy in the market. In former USSR like today's Russia, Ukraine, you get the dehulled millet in packet. They primarily use as a porridge, either with some milk or no milk, and they put some kind of pumpkin here, shredded pumpkin. That's the one recipe from, this is from Russia, and similar situation in Ukraine. This is some of the millet in Asia, in Nepal, China. This is millet rice. This is the millet kind of flat bread from India. These are all made from prosomillet. So prosomillet makes really a delicious food. If you think the prosomillet is the bird feed, that's wrong. Here is the prosomillet for alcoholic beverages. This is the traditional way to ferment the prosomillet for beverages in Nepal. I forgot the name, I think Dungu or something. These are all bamboo internode. That's the way they ferment, and then they drink that fermented prosomillet milk. That's the traditional drink. In conclusion, I would say we started working some of the modern breeding aspects of prosomillet. Our program is the only program who are doing little bit of agronomic research of prosomillet for the semi-arid production system. The biomedical research from not from us but from other part like from Japan, they're on many other places, they are showing it is really human healthy. We need to develop some ready to eat prosomillet based food for our fast lifestyle. Traditionally in China or Russia or Asia, they use prosomillet for human food, but they start from the scratch. Is a long cooking process. Our today's 21st century's our fast lifestyle is probably not compatible that way. So in order to expand prosomillet, such a beautiful climate resilient cereals, in order to incorporate more and more in our food chain, we need to develop ready to eat food products. As I indicated, my program is the only breeding program, genetic improvement program for prosomillet in Western Hemisphere. Public-private partnerships essential for all these things, not only breeding, for product development. Need to collaborate with seed companies, need to collaborate with big food industry. Public awareness, is extremely essential. People may think cereal-based food means only bread or rice, but that's not the case. People need to know the proso millet is healthy, and more they eat, more it will be grown, it will be better for our future climate sustainability. We need more farmers' engagement. Farmers need to come to the network, which is researchers and industry. So farmers needs to come there so that they know the whole picture. And last but not least, we need a global engagement. Why I'm telling that? Not many research program of proso millet or in general millet, except probably pearl millet, are there all over the world. As you see this map, I kind of try to depict that. There is, my program is the probably only proso millet program in the world who are really truly doing some proso millet breeding, like vertical breeding, trying to develop modern genetics tools. We are trying to develop some agronomic production tools. We are trying to develop some food related research, but there is no other program of our kind that big and that comprehensive anywhere in the world. Nevertheless, there are few programs who are working on some aspect of proso millet. For example, in India, in Hyderabad, central India, they have an ICAR, they have a millet research institute. They are working on some aspect of genetic improvement of proso millet. In Yangling in China, northwestern part of China, they are the another big proso millet research, but not too much of breeding as of today. And then there is also small breeding program, not breeding program, but proso millet research program in Italy, in France, in Austria and Switzerland. They are trying to develop revive the proso millet. Poland revive the proso millet, which is almost like lost grain of 20th century, because proso millet was popular in that part of Europe, and slowly it is replaced by modern crop like corn, modern wheat varieties. But now, because of the climate change awareness and some of the human health aspect, they are realizing that we need to revive our ancient grains, which our parents, our grandparents used to use. So they're trying to revive that. So these are the few research program across the world. So global engagement is extremely essential if we want to develop, make some significant progress in shorter possible time. My team, these are my alternative crops breeding tool team, my technician here, then collaborators, James Snobel, Devin Rose, and Yu Zhang from my University of Nebraska. Some of the possible funding here I noted. And with that, I'll conclude my talk. And as I told at the very beginning, my email address, dsantra2@unl.edu. If you have any question, you can email me, and then I'll try to respond you. And I do not know if there is any place for question answer at this session. Otherwise, probably that is the end of my talk.
Thank you, Dr. Deepak, for your presentation. And yeah, it was really informative and worth waiting for it. So yeah, do we have any questions? And in the meantime, I really need to apologize for all the inconveniences caused due to the time differences. And I'm really sorry for disturbing you early in the morning.
No, that's fine. And finally, we made it. And probably it is not only your fault, it's partly my fault. I should have paid more attention to the time. It is written in British time, John. Yes. So partly my fault also, but it worked out at the end. I'm glad I could present it.
Yeah, that's great. Thanks a lot for understanding too. So yeah, and for the next time, we really promise that we'll definitely make it up for the US timing for sure. So,
yeah, as long as the New York time zone, that would be okay. But anyway,
the main concern is that understanding the Chinese and the South Korean time. It's almost like it's around 3 a.m. in the morning. So if we'll be starting their presentations now, so it will take a lot of time. So that's the reason we had to.
I will accommodate next time. It was partly my fault, as I told. And when you have an audience all over the world, Somebody has to sacrifice, may have to give presentation at 1 o'clock in the morning or 3 o'clock in the morning, that kind of things. Nothing we can avoid that. So I understand that.
Thank you. Thanks a lot for that. So any questions? I guess there are no questions. So yeah, thanks a lot. It was really a neat presentation. Thank you.
Thank you.
Have a great day ahead.
Sure. Thanks. Bye.
Thank you. So with this, we have come to the end of our today's sessions. And thank you one and all for all of your time and your presentations and all of your research details and everything. Each and everything was really informative and very impressive. So if any one of you have missed any of the presentations, so we'll definitely be posting this in a link, and like the video will be shared to all of you, and we'll be sending an email to you. So thanks a lot, and we really welcome any of your suggestions or your feedbacks regarding the conference. So we'll definitely keep you updated about the next webinars coming up, and we really hope to meet you all at a conference, maybe in organized in any part of the world. So yeah, we really hope to meet you all in person after this pandemic ends and with everything and everyone vaccinated and everything. So thank you. Thank you one and all for your time and your patience. Sure. So as Dr. Deepak has sent his email, so yeah, for any questions, you can contact Dr. Deepak. Thank you. Thanks a lot. Have a great day, everyone.
Bye.
Thank you.